Now we have the round table where we can do the Q&A. Thank you.
So thank you very much. I would like to invite some people here for sitting in the front, for answering some questions, if any questions, and comment on some of the questions that we already have touched some of them. Christopher, please, from University of Caen, was the Vice President of Research. I don't know, Olari is here? Olari? No? Then we have Professor Jack Baloso, Ivana, Takashi, please.
Yeah.
Yeah, you're already here. And Walt is already here. Mats, please. And Manjit. So we have two moderators, Angelica and Karim. Angelica, there is I think it's, it's up to you.
No, it's better. So we can also give this one.
Yes, I think we need to have it. So I ask you first one. I think I would like to ask the first question, then I will give you the microphone. And this is to our Vice President, Christophe. I would like, actually after hearing all the discussion today, particularly in radiobiology, I would like to hear your view.
Yeah, the microphone.
Your
the microphone.
Oh, that's on the
No, it's working. It is working. Yep.
So I would like to ask you, as a representative for the Caen University, so how do you think that the university can facilitate and support the research program that we discussed? And some of those perhaps will be taken up here and start to do, and some we already have, and we will continue with that within the Hadrontherapy. And my second question is about the, for any research program, particularly where we are connected to the university, and education should be also connected to. So what is your point of view considering the whether a kind of course or master program or university will support us to set up such a course within the Hadrontherapy for whether biologists or medical doctors, medical physicists, to facilitate or to educate the next generation of the researcher, let's say, in this case.
Okay, I understood that we are a bit under pressure with time, so I will try to summarize my answers with quick comments. So the first question is how to support your research activities. Of course, there is many ongoing supports that we could offer for mobilities. I think you have a fantastic community here showing that you have already collaboration between all of you. Just know that we have many opportunities for invited professors. You want to come to spend more time to come. We will be happy to welcome you. You will have more time also to enjoy Normandy. So apart from that, there is within our research council some support that we could offer to create international laboratory partnership in order to foster more direct collaboration between one or two universities. But I think the more the particular program I would like to stress today is the excellence program that the university have attracted over the last few years. It's called the KISA project, and it is really one. We are one of the few universities who have dedicated such a project to support our research community. Within the next 6 years, we will spend more than 20 million euros in order to really boost the research activities of our community in some specific fields where we would like to have a sort of signatures of our research in Caen. Among those, there is 8 different topics that we think is really at the top level at the moment. One is, of course, nuclear, which include also nuclear research, nuclear physics, but also nuclear medicines. And there will be dedicated funds in order to support these research activities. Now there is also another topic where we think we have fantastic capacities within our different research groups, and we'd like to boost this activity to reach European activities, I think, in order for us to apply with some of our colleagues to European funding. And in this case, it is innovation in health. So in both of those fields, I think there is possibilities to support research activities in Caen, but also to reach the levels of competitiveness that we should have at the European level. And I think from what we've just seen and from what I've heard today, I think we are, at the moment, really, we have all the forces within the university and with the collaborators, of course, in the Baclesse centers, but also in CYCLHAD. So I think that we have everything ready here. Now we just have to connect our different forces in order to reach these competitive activities. Now coming back to the second questions concerning education, I think you have already the network. So there is many possibilities. You know that Caen, especially in nuclear physics, had already engaged a possibility to develop an Erasmus Mundus research masters. And I think that's maybe a topic that we could address apart from this nuclear physics education program, that we go more on the radiobiology. We have also conducted and supported these activities over the last few years. We have already 4 ongoing Erasmus programs in Caen, and we'll be more than happy to support any wishes of our colleagues to develop such courses with European collaborators. And I think that's really with the objective to train future generation of students equipped with the sufficient competencies to develop our research activities, and also to go in all of your laboratories abroad, maybe for future education. So I think we will be more than happy to develop these together with our colleagues in the different universities.
Thank you very much. Thank you. I just wanted to say that the discussion is open. I just had the first question. So please feel free to comment, to add something, and also important that you mention your name and affiliation. I already present you, but I think if you take the microphone.
So I'm Mats Ringdahl from Stockholm University. I'm a professor emeritus in radiation biology. So I just realized that I started 50 years ago with my PhD exam. So I have some experience, and I have been, this is a presentation, so I leave my questions.
So if it's a question of presentation, you have seen me this morning presenting some topics about clinical research in the frame of Étoile project. So I'm Jacques Balosso, radiation oncologist from the University Hospital of Grenoble, working very tightly with the university and the Hospital of Lyon for this project.
Manjit Desange. We talked about networks. I've actually been working on the European network ENLIGHT, which started a long time ago, and I just feel like we are reliving when we started, when the PIM study had just finished. And a lot of this was really trying to establish what was needed in terms of research, education, training. And we spent quite some time trying to get funding for training the future generation, and then funding for doing what the basic research was. And then the final project under this, which HITRI Plus is a continuation of, was ULIS, which was really then doing 3 pillars: clinical access, beam access, networking, and of course research and training. And I just get the feel that we are deja vu and we need to do some of this anyway. So that's enough for me. CERN, and now I'm also a little bit retired from CERN, but a visiting professor at Oxford, but carry on doing ENLIGHT, of course.
So you heard my talk recently. I'm Ivana Dogic from Heidelberg, from HIT. and I'm a group leader there from particle radiobiology. And I also mentioned in my talk, so we are having a very nice collaboration with a group in Paris at Marie Curie Institute. And I think it is nice to start every collaboration with writing a common grant that Dave gave. So this is a German funding society. It's quite straightforward, easy. So as I said before, we are open to have students which we could co-supervise together on the master level, PhD level. So yeah, I think this is something that should be advertised a little bit more and more transparent.
I'm Walter Zinganelli, clinical radiobiology group leader, and at GSI, and essentially, I mean, I share exactly the same opinion and idea. We are also very open to collaboration. We have a lot of collaborations. I mean, without collaboration, we could not honestly survive right now. I mean, it's important to stay always in contact with the other institute. It's always important to do some training, and of course are important funding, because when you want to do collaboration, it's very important that there is someone that can give you money for consumables, but also for guesting scientists, that in our case, I mean, are more than welcome.
I'm Baclesse from Japan, so in Japan, a little bit far from the European countries. So, but still, I also agree this collaboration is a good way for, especially for this particle therapy, because fighting or competitions, for sometimes good, but for keeping such high qualities and also keep the nice experiment to connect it to the clinical output. So that collaboration is very important to keeping all these activities.
Yeah, I'm arising a quite hot topic for people working with carbon beams, the cost of the beams. Because, Walter, you were talking about consumables and cost for hosting people, but that is quite, I mean, we have to face to the fact that beams are really costly. We have seen that in Japan they have to reduce the number of beam time for experiments due to the cost. So in your experience, apart from what I presented with ITRI+, that is in a short time window, how can you deal with the cost of the beam, and how in your facility do you manage the cost of the...
Should I start? Okay. So at GSI, as I said, we are user facility. So in principle, we have to provide beam time to external users. But essentially, external users are not paying for the beam time. So we have like some money. So GSI is like 90% federal, paid by the German state, 10% by the state of Hesse. And then essentially are the taxpayers that are paying the beam time. So there is a commission that approve the project, and that project they have like the beam time granted. Okay. For the HIBER instead is, as I said, we are ground-based facility of ESA. But also in this case, ESA is not really paying the hours of beam time. They just provide some funding for the, let's say, to support, let's say, those projects that are selected. So that's... Then we go in that direction.
Yeah, so for HIT and for the experiments at HIT, we have a lot of external groups who have funding, and as I said, it's relatively easy, I would say, to get the funding for the research that is sensible. So for this, we have to do the pre-selections of the projects, which would be allowed actually to do the experiments at HIT. And actually we had a system for doing this, and now we are building an online platform where the people who are coming from external and want to do their independent research there can apply. And then we will see and estimate the cost. So I have to say the costs got really expensive in the last few years because of the electricity costs. So just as an approximation, so the HIT uses electricity per month, something like a small town nearby Dossenheim. That is some 10,000, 15,000 people, if I'm not wrong. So it's a lot of electricity for this. And what we say to all the external users is that they have really to plan their biological experiments really, really well. So it's something else than when doing the physics experiments, because the most of the night shift, because the experiments are in the night, are preparation of the samples, not necessarily beam time. So the pure netto beam time is much, much, much shorter than what one thinks. So therefore, it is really important that the people who are interested in it prepare themselves and to have the defined project. And as mentioned in the talk by Dr. Fossati, is that the research has to be sensible and clinically sensible and clinically relevant. On the other hand, we have also collaborations where we do the projects together, and such a beam time is then funded from our side, so basically from our internal budget, and this is also going into evaluation when the people apply for such a collaboration. So there are parts, we have a lot of groups that are coming to work with us. It is just a way of being well organized and prepared and applying for a beam time timely, effective, because it can take a couple of months to go through all procedures.
I'm not sure that I can tell you much about beam time. I did know until about 5 years ago there was a huge shortage of access to beam time. And in order to be able to do a range of experiments, which we've been hearing today, and that was one of the reasons, just like you are saying, Walter, for GSI, being at CERN, we wanted to try to provide a biomedical facility for anybody who could come and use this beam time. But it's not just the beam time, it's also facility, it's all the infrastructure. Biology experiments are expensive, but as I said, it happened certainly, so I can't say much about it because CERN, of course, the primary mandate of CERN is high energy physics. So, it depends on the mood of the management at that time. So we were not able to do that, but I'm hoping to hear that we have better access for beam time now, but certainly 5 years ago, there was a huge shortage of beam time. And it's clear to me, listening to everything we've heard today and we will hear tomorrow, there's a lot of research to be done, there's a lot of experiments and different types of things, and not just biology, but also development of instrumentation, all of this, and it needs that, and it needs resources. So we have to think about how we're going to make that feasible. And as Angelica was saying, okay, so GSI and CERN, we have government funding and all of this, but clinical facilities, they have lots of challenges. So I think we do have to think about how to be able to collaborate and work together and look for funding. And what we did, started 20 years ago, was go to the European Union. But I think we need both national and European and beyond. This is what I'm thinking. And we have to be very strategic. And you're right, Ivana, that everything has to be planned very well. So it means collaboration. And I only, as a little bit of a joke, when we mentioned, we named our network ENLIGHT, the European Network for Light Ion Hadrontherapy. And I say this, and I was saying, my particle clinical physics was Hadron. Why can't we call it particle therapy? And somebody like Ugo Maldi saying, yeah, but electrons and photons are particles, so why can't we call it? So just the idea that it's a really multidisciplinary collaboration that's needed for hadron therapy or particle therapy, whatever you want to call it. So, and it shows you the flavor and the needs. So I think everybody's happy, we want to move forward, but we do need resources and expertise and the future generation, because it's not the machines that are going to treat patients, it's going to be people who do this. And we need the research to be able to see which are the challenges we have to meet.
Maybe I could give an overview of the French situation. Some years ago, through an infrastructure called France Hadron, we had as one of the first concerns to make possible the access to beam time and to finance this beam time. And in this structure, quite a lot of money has been spent for that. And I should say that the situation is completely different, not completely, but is different through the different institutions in France. There's not a general scheme. To make things shorter, we could say that if you are involved in a strong collaboration with also local interest in the research, certainly external users could have free access to the beam. This is the case in Nice, it could be the case in Institut Curie, it could be the case in Strasbourg, it could be the case also maybe in Nantes. But if it's pure external users, the beam will be costly. And the cost level has raised these last years, of course. It is only the time of use of the beam, not the present time in the institution. So that's the general situation presently in France. And of course, the Normandy project of CYCLHAD and ARCAD is mainly answer to that shortage of beam time and the possibility to host for different time, some days, some weeks, some months, external user, external experimentator in the CYCLHAD building and have access to the protons. It's possible now since December 21, not as a staying experimentator, but coming from outside using the proton beam. It could be in some years possible for carbon ions, and the final goal is to establish laboratories in the building, as you have seen for CNAO or for Medostro. It's the same spirit in a way, to make possible to make good experiments, not having the concern of going back home at the end of the irradiation with some samples which are difficult to handle and so on, but to do really the experiment locally and to have good results. So that's the goal. But this way, the beam will not be free, and we have concern about the price level of this beam, and we hope that the quality and the possibility to have 100% efficient experiment will counterbalance the cost of the beam, because this cost will be quite heavy. So on the other hand, when you are buying a biological reactive, you are spending a lot of money for some things. And while the irradiation with so sophisticated means could be free, that's not possible anymore. So in your demand, in your grant demand, you have to specify that some money will be used to buy access to irradiation. Of course, you have some silly rules that these costs are not eligible. We have that in France, for instance. So that should change. We should convince, maybe through universities and through faculty body, that this matter is really costly and should be recognized as an expense when you are making a grant application and things like that. That's very important. We still have some problem about that. I would like to make a comment also on models we could have to deal with this experimentation. We have spoken a lot about ex vivo model, in vitro models, and about in vivo models, small animals, large animals, as said Piero Fossati. With the rising application, clinical application of hadrontherapy, we have more and more clinical questions which couldn't be solved by ex vivo models. For instance, the sequence between associated treatments, when you are doing photon or proton and carbon ion, what should be the good sequence? The same time, one before the other, and so on. That's a simple example, but there's a lot of others. The combined modalities with chemotherapy, with systemic medication, with immunotherapy and so on, all that will need animal models. And I like your intervention, Piero. You said that finally on small animal, when you are injected the tumor, you don't give that tumor the chance to develop the right way, the natural way. And you spoke about the pet patient. That is a very good way. We are doing that in Grenoble since 10 years maybe now. And it's really a good way to treat real tumors. Of course, it has not the homogeneity of an experimental tumors, but some animals have very regular tumors which are very similar. And if you have a very good deal with a community of veterinary, you could have this patientèle, if I may say, and it's really something you can do with a strong input, a lot of animals, a lot of a deep implication of the proprietary. These people are wanted to treat their companion, and really it's something we can do, and to begin for some months with this type of patient in a new facility is certainly a very good way to proceed, really a very good way to proceed. And veterinary in France and the veterinary clinic are quite frequent. There's the one very close to CYCLHAD, by the way, and it's nice collaboration we could do. And by the way, in the French network of preclinical research in radiotherapy, we have several teams of veterinary in radiotranslation. So it's really possible.
Can I just,
Well,
just
sorry.
wanted to say something about
Yep.
the animals, because this is flash, but it doesn't matter what it is. But Marie-Catherine Voisinine in Lausanne did exactly that with cat and dog patients. The owners are very happy because they're getting treatment, and she's happy because she's getting the examples, and of course the pets are being treated. So just to say, there's actually a live example for the last 5 or 6 years that Marie-Catherine Voisinine is doing this actually very effectively. Sorry.
Here in Caen, we have a special team. We have a very nice animal housing facility at CIRT. We have the marmoset. Marmoset, also known as Wistiti. So some little monkeys that look like, human brain, for example.
Well, no.
Maybe a bit,
Well, just be careful because it depends also by the national laws. You are not always allowed to treat animals in a clinical human facility. So just be careful and consider that it's a very nice and surely spontaneous tumor are totally a better model, but just be careful because could not be so straightforward.
Can I? Should I? It's okay?
We are running out of time.
Okay, so just a short question. So the idea for us in Caen for radiation biologists is to benefit from your experience. And so do you have advice for the facility, for equipment and everything, so we can avoid to lose time? And so I heard from Angelica that you said that animal facility should be near the lab. And so these kind of things that perhaps we didn't think about.
May I make a comment? I'm a radiobiologist, so I am the user of the beams, and I understand it's very expensive. I have learned today even electricity is a problem. But as a user, my experience is that you need quite a good grants, and you need a lot of collaboration. And the problem I've realized in Scandinavia is that the funding for basic research in radiobiology is going down. It's a general problem, and this is also reflecting into the clinic, because like if I mention Karolinska Institute, it's a high-ranked hospital, and they are lacking the basic knowledge among the radio-oncologists. And it's being realized as a problem, and they are trying to solve it by different kind of collaborations. But at that hospital, this part of radiation knowledge is diminishing. So I don't know what is the experience here, but and in Europe I have contacts with a network concerned about radiation protection research. They are facing the same problem. Radiation biology for the future is at the risk. So I think among the things we should discuss also the funding for experimental part of the research for high-LET radiations. And I think also as a very important part is the education and training program for the future. Because you are young in my view, but still, we need the new generations to come, and I think you mentioned the education program, and it's a very important part of the European network that we can collaborate within different kinds of radiation biology problems. So I think it should be included in the discussions about how the future use of new radiation technologies and so on.
Can I comment?
Yeah.
One more comment, and then we have to.
So I'll just try to comment this really fast. So when making radiation biology lab, it's actually making any kind of biology lab. So there is nothing special. So you should not reduce, to my opinion, your biological cancer translational research to the word radio, because with the technology of radiation, the biological techniques for imaging, single cell sequencing, flow cytometry, I don't know how many there are that are going advanced. And if you reduce yourself to doing classical radiobiological methods as clonogenic assay forever and gamma H2 foci, it's not going to bring you grants that you need for the beam time. So I think you should focus just having as any developed possibly biological lab that will be in a close proximity, as you mentioned, that is very important to the radiation source, ideally with the animal facility, if not getting collaborations as much as possible for something like this. But again, coming back to what was previous said, the first thing that this research has to win the grant, and to win a grant it has to be innovative and helping the humanity in general, and not be just research per se, just for having fun because you are interested in a certain pathway. No, it has to be really relevant, and it has to help people, and not economically relevant for the universities and overheads, but actually relevant for humanity. So this is, I think, as every biologist, cell biologist, oncologist would tell you, but yeah, just try not to reduce yourself to radiation research purely.
Yeah, I mean, I agree on. Sorry, one second. I mean, I agree on that. You know, I'm sure that we are moving in a new era of radiobiology. So it's not the classical radiobiology, but now it's radiobiology of the omics. And then, I mean, it's important, it's necessary that the radiobiology lab has more sophisticated device, because otherwise, as Ivana said, you are staying always with the old, let's say, kind of radiobiology that of course was good. I mean, we all work using that radiobiology, but nowadays we have to move forward. And in my opinion, I mean, the animals and an animal facility, it's necessary. I understand your point, and I agree with you. I mean, it would be a lot better if you could use like natural developing tumor and so on. But there are a couple of points. First of all, I think with the regulation nowadays, it's very, very difficult to transform your KV radiation room in a facility where you can treat like pet animals. And the second point is, I mean, most of the time when we do animal experiment, honestly speaking, sometimes we also try things that are completely new. And the question is, are you sure that the owner of a dog or a cat will give you the possibility to try something that is completely new? I mean, that's also important. I mean, with them, you should use a treatment that is already known to produce some effect. Or at least you have to justify and say, okay, we are trying something completely new, and we give you, I don't know, 30% of possibility that your animal will be okay after that. It's absolutely difficult to do this kind of step, but I agree with you. And the other point would be like to have animals that develop the tumor. I mean, there are mice and rats that develop tumors spontaneously, but then there is another problem. You have to coordinate the point when the animal develops the tumor with the radiation. And you have to hope that all the animals develop the tumor in the same time. Otherwise, you will irradiate one animal per month. So that's, in my opinion, we still need the animal facility. I mean, the old animal facility, rats and mice.
Just Piero. Oh.
She was asking
the
about
last. It's the last comment, because we have to.
So just maybe the optimal sample to show what you want to show in your biology experiment is whatever, but my advice would be always include another sample that you can also check in patients. So this is quite fundamental from my point of view. You will not get the lung tissue, you will not get the bio, but you can get the plasma. So it's a big pity when you do mice experiments and you don't have the same. And say, by the way, you find it also in plasma, even though not so nicely as in the tissue, and then you can correlate with the patient treated in a similar way.
No, that's sure. Okay, I think we have to.
Thank you all. Yes, thank you for the discussion. I'm afraid we have to stop here. Thank you very much.